In order to determine what pigments have the ability to absorb which wavelengths of light, we can simply use a spectrophotometer to help us out. So we've used a spectrophotometer in lab, and we've got in the top part of this slide here the internal workings of one of these spectrophotometers, right? So we've got a white light bulb inside, and that shines the full spectrum of white light onto a prism. Prisms, remember, will bend light, and so they have the ability to separate out all the different wavelengths of that white light, from the reds all the way down to the violets. Once you've separated out all of the wavelengths of light, then the wavelength selector here will choose only one wavelength of light to allow to pass through this hole in the center. So in this case, we've got green light green light has been given a chance to pass through into the sample chamber here, where we've got a solution of chlorophyll in a test tube. Now we know already that chlorophyll is not good at absorbing green light, right? So if the chlorophyll is one of the main photosynthetic pigments in leaves, and the leaves appear green to us, this is because that green light was not absorbed by the chlorophyll and is either reflected or transmitted through the leaf and makes its way to our eyes so that we can visualize it. So as the spectrophotometer shines that green light through the chlorophyll here, you'll notice that very little, if any, of that light was absorbed. The majority of the light gets then detected by that detector within the spectrophotometer, and the spectrophotometer display should show close to 100% transmittance, indicating very low absorbance of light. We can do this for other wavelengths of light. For example, moving the wavelength selector knob here to allow blue light to pass through the chlorophyll sample. Chlorophyll is really good at absorbing blue light, and so the majority of that light energy will be absorbed by the chlorophyll molecules. Very little of that light will make its way to the detector, and the spectrophotometer will show close to 0% transmittance, indicating that the majority of that light was actually absorbed by the pigment molecules. Now, we don't just have to measure the absorption of green light or blue light. We can do this for any wavelength of light within the visible spectrum. And if we were to do that, we could graph what is called an absorption spectra. That's at the top half of this slide. So on the x-axis, we end up plotting the different wavelengths of light from the 400s to the 700s, and on the y-axis, we can plot the respective absorption readings from the spectrophotometer. So you can tell we've got three different kinds of pigments here. Chlorophyll A, for example, is this blue-green pigment, and it's really good at absorbing the violet light and into the blue light. It drops off in the greens and then comes back a little bit here in the red-orange. And so you can see how different pigments will have different absorption capabilities of different wavelengths of light. Now, you can imagine that an organism that, let's say, has all of these pigments, the chlorophyll A, B, and the carotenoids, any area along the visible spectrum where there's high absorbance of light by these pigments, you'd expect that there's a lot of energy that gets funneled into driving photosynthesis. And so what scientists can do is they can record action spectra, where photosynthetic organisms are given different wavelengths of light, and we ask the question, how well can that organism use that particular wavelength of light to perform photosynthesis? The way we measure photosynthetic output is by measuring how much oxygen is produced. So you'd expect an organism that has these pigments, depending on where they have the ability to absorb a lot of light energy, you would expect there to be a high rate of photosynthesis measured by a large production of oxygen. On the other hand, those wavelengths of light that cannot be absorbed by the pigment molecules, you would expect a decrease in photosynthetic output. Again, less oxygen will be produced. Let's talk about some of those different pigments that we've got. So we've got chlorophyll A. This is going to be the main photosynthetic pigment. This is the one that does the majority of the absorbing of light energy to send into photosynthesis. Then we've got accessory pigments. Chlorophyll B, it also helps to absorb light energy for photosynthesis. Maybe not as much as chlorophyll A, but it helps. It actually also broadens the spectrum of light energy that can be used for photosynthesis. So if we take a look at the absorption spectra down here, chlorophyll A, for example, only goes maybe into the deep blues. But anything past that, and it really can't absorb much of the other wavelengths of light in the blue range. So chlorophyll B, if you add that to the mix, you'll notice that its peak is actually well into the blue range, and so that allows an organism having both chlorophyll A and chlorophyll B a broader range of that visible light to use for photosynthesis. The carotenoids are also accessory pigments. The carotenoids, while they don't contribute as much to pushing energy directly into photosynthesis, they function more in a protective role. So they tend to absorb any excessive light that might otherwise damage the chlorophyll pigments. And those are the ones that are responsible for photosynthesis. I like to think of the carotenoids more like sunscreen, right? We put on sunscreen to protect us from the UV light that comes from, or UV rays that come from the sun, so that we don't cause damage to our cells and their DNA. The carotenoids are very similar to that. So they absorb that extra energy that hits the pigment molecules, that hits the chloroplasts, and help to sort of funnel that away from the chlorophyll A and B, keeping them in good working order. All right, so another thing that we really haven't covered yet, I mean, we know that different pigments absorb different wavelengths of light, but we haven't really talked about actually how this process takes place. And so to understand this, we have to really go back to chapter 2 and a little bit of chemistry to make sure that we're all sort of on the same page. So here I've got drawn an electron, an atom and the different electron shells. So in the center right here, we've got the atomic nucleus, and then we've got the concentric circles showing you the different energy shells, right? So you've got your first energy shell, your second energy shell, and then the third one on the outside. And in chapter 2, we talk about how electrons, they can move from one energy shell to the next, right? So this little blue guy can move from the first energy shell here to the second energy shell, or one electron from the first energy shell can move to the third, and so on and so forth. But the thing is that if an electron wants to move between these energy shells, it needs to either absorb or give up the exact amount of energy that's the difference between those two shells. Right, so if an electron, for example, in the second shell here has 2 units of energy and it wants to move up here to the third shell and needs 3 units of energy, it's got to absorb exactly one more unit of energy to be able to go up to that higher energy shell. So remember that photons, right, are discrete packets of this light energy, and each photon, that's a different wavelength of light, will have a different amount of energy associated with it. And so the atoms within the chlorophyll molecules, right, their electrons can move to a higher energy shell if they get hit by a photon of light that's got the right amount of energy. If it's the right amount of energy to boost them to that higher energy shell, then that electron will absorb that energy, jump to the what we call excited state, and the photon of light gets essentially absorbed when the electron does this. So let's take a look at the top half of this figure. Here we've got a chlorophyll molecule, and it's kind of like a graph, right? So progress of reaction here goes from left to right on the x-axis, and then the energy of the electron is shown here on the y-axis. So if the chlorophyll molecule here is hit by a photon of light, and it's the right amount of light, meaning the right wavelength of light, that has the right amount of energy to take one of the electrons and excite it to the excited state, that electron will absorb that photon of light. It jumps up here to the excited state. But think about what we understand in terms of energy versus stability. Anything that has a lot of energy, it has very low stability. So that electron in the excited state, yeah, it's got a lot of energy that it just absorbed, but it is very unstable. So it doesn't want to be there in the excited state. It wants to go back down to the ground state. When it does that, right, it will, once it reaches the ground state, it will reach maximum stability, but it gives up all the energy that it had just absorbed. Now the energy gets released back. The majority of the energy gets released back as heat, but then some of it can be used to release another photon. We call this photon that gets released fluorescence. Now there's a difference though between this photon that gets released and the one that initially excited the electron. They are not of the same amount of energy. Right? This original photon had a lot of energy that was given to the electron to reach the excited state, but that same amount of energy, when the electron drops back down to the ground state, not all of it can be released as a photon of the same wavelength, right? It's the energy that gets released, part of it gets lost as heat, and so the remaining energy left for the photon is going to produce a photon of lesser energy than the original photon that actually excited the electron in the first place. And so we can show this using chlorophyll molecules. If you were to take chlorophyll and look at it in the dark, right? In the dark, on its own, it would be black, right? You wouldn't see anything. But if we were to take a UV light flashlight, right? So UV light to us almost looks bluish in color because it's on that end of the spectrum where we have our short wavelength, high frequency, high energy light. Maybe let's go back a slide and just take a look, right? So here we've got our visible light. Ultraviolet light would be over here, right? So into the 300s. You'll notice that if we were to shine UV light on, let's say, chlorophyll molecules. If you take a look at chlorophyll A here and its absorption spectrum, it's not just going to immediately drop off here in the high 300s. It's going to slowly taper off, or you can imagine the slope here of the line. And so chlorophyll A has the ability to absorb UV light. And so if we take a flask of chlorophyll in the dark, shine UV light on it, those UV light energy packets will be absorbed by the chlorophyll A electrons, sending them to the excited state. They freak out, don't want to be there, drop back down to the ground state, and they re-emit that energy. The majority of the energy is emitted as heat, so eventually that chlorophyll flask would heat up a little bit, and the rest of it will be emitted as photons of light that have far less energy. Right? So anything down here on this towards the right side of the visible light spectrum would include photons that have less light energy. And so if we were to do this and excite our chlorophyll electrons with UV light, the photon that gets re-emitted as the electrons drop back down to the ground state is a red photon, or a photon of red light. And so you would see that entire flask of chlorophyll glowing red or fluorescing red in that flask, which is definitely kind of cool to see.